Hello students, welcome to EPG Pathshala. I am Gayatri Ramakrishna from Institute of Liver and Biliary Sciences, and today we are going to discuss the module Cancer Stem Cells from the paper Molecular Therapeutics. Cancer stem cells are also referred as tumor initiating cells. Just as the normal stem cells, these cells are crucial for the development, regeneration, while the cancer stem cells underlie the tumor formation and its maintenance. Rare cancer cells or the cancer stem cells have been isolated from a number of human tumors, including the hematopoietic, the brain, the colon, and the breast cancer. An important question is, how are cancer stem cells derived? Invariably, they arise from the normal stem cell. Cancer is increasingly being viewed as a stem cell disease, which in turn is attributed to uncontrolled proliferation of a minority of stem cells with stem cell-like properties, and its possible derivation from the normal tissue stem cells. The activity of the stem cell is tightly controlled through the various signal transduction pathway. This raises the question how a normal regulation of the stem cell deviates towards the genesis of a cancer stem cell. This is best understood by the fact that there is a close association between the normal tissue repair activity and the process of the cancer. The underlying feature is the process of cellular proliferation itself. In fact, during tissue regeneration, the normal cellular proliferation is regulated, and the repair is completed and the proliferation stops. However, if the injury is chronic and persistent, the same tissue regeneration signals leads to an uncontrolled stem cell renewal in a chronic injury, thereby transforming a normal stem cell into a cancer stem cell, whereby accumulation of the oncogenic insults. This results in a cancerous growth. This module will cover how to identify the cancer stem cells. Further, it will help in understanding the important implications of the cancer stem cells for the cancer therapeutics. After completing this module, you should be able to understand what the stem cells are, what are their basic property, how the normal stem cell differentiate from a cancer stem cell, what are the basic characteristics of a cancer stem cell, the role of stem cells in normal regeneration and in the process of cancer, the pathways which are common and how they get deregulated during development towards the cancer stem cells, and briefly the module will also cover parts on the markers which can help in distinguishing a stem cell and a cancer stem cell. This module will help you in learning the concept of normal adult stem cell vis-a-vis a cancer stem cell. Cancer stem cells are defined as those cells within a tumor that can self-renew and drive the process of tumorigenesis. So now let us understand the definition of a normal and a cancer stem cell. A normal stem cell is defined as a rare cell within the organ that can self-renew and give rise to diverse cell types within the organ to drive the process of organogenesis. On the other hand, the cancer stem cells are also a type of rare tumor cell that can self-renew and give rise to phenotypically diverse tumor cell population to drive the process of tumorigenesis or cancer. Let us understand the role of normal and cancer stem cells in context of the normal regenerative process as well as the cancer. The example that has been picked here is from the process of liver regeneration and also how the hepatocellular carcinogenesis is driven. Following insult of the liver by viral infection, drug, or alcohol-related injuries, the bulk of the hepatocyte gets damaged, but the few remaining hepatocytes then divide and the process of liver regeneration occurs. In extreme conditions, when the normal hepatocyte division is blocked, then stem cells come into action, as is shown in point number 2. And the liver adult or adult stem cells or the hepatic progenitors help in the process of liver regeneration. In certain instances, due to activation of oncogene or tumor suppressors, the normal stem cell can convert into a tumor initiating or a cancer stem cell, as shown in point 3. And then starts the clonal proliferation of this tumor initiating cell or the cancer stem cell as it is called. This leads to formation of a malignant cancer type. To understand this, we need to first understand how the normal stem cells divide, and this is shown in the next slide. Let us now understand the properties of the stem cell. The two hallmark features of any normal stem cell are the properties of self-renewal and differentiation. Self-renewal means they are capable of dividing and renewing themselves for a long period, and at the same time retaining their unspecialized stem cell characteristics. In case of a need, the daughter stem cell can differentiate into an effector cell, which can then acquire specialized functions, such as that of a hepatocyte, cardiac cell, lung cell, etc. What determines the self-renewal versus the differentiation fate? To understand this, we need to understand the cell division in the stem cell. The fate of a stem cell is actually determined whether the division is symmetrical or asymmetric. In a symmetric cell division, it gives rise to two similar cells retaining their original stem cell property. However, when division is asymmetric, it results in two different cell populations, one retaining the original stem cell characteristic and the other becomes the effector cell, which then differentiates into various cell types. Thus, the type of division is very important to understand the process of differentiation fate. Let us now understand what are the cancer stem cells and what is their characteristics. Cancer stem cells are rare cells with characteristic property of the stem cell. That is, they show self-renewal and also the process of differentiation. However, unlike the normal adult stem cell, the cancer stem cells or CSCs show uncontrolled cellular division, giving rise to a tumor cell mass, which has the properties of metastasis and invasion. Further, the cancer stem cells are resistant to chemotherapy and radiation therapy. How did the concept of cancer stem cell arise? In the earlier days, the most popular consensus, as shown in the part 1, was that every cell has an equal potential to form a cancer. For instance, in this image 1, there are a number of balls which are all differently colored. If all of them have a property to form a tumor, you will end up having a heterogeneous mass consisting of all the different colored cells. However, a series of elegant experiments in blood cancer revealed that not all cells can form cancer, and this is shown in figure 2. As is shown, only a rare cell, for instance here, the red ball, can form cancer, not the other types of the cells. This led to the concept that there is a presence of a rare tumor-initiating cell which can lead to the formation of a cancer, and this rare tumor-initiating cell is often termed as the cancer stem cell or the CSC. Now let us understand the origin of the cancer on the basis of the two popular theories. The two popular theories are the stochastic model and the hierarchical model. As was explained in the previous slide, As per the stochastic model, each cell has a potency to divide uncontrollably and can give rise to a distinct population of uncontrolled growth mass or the tumor. In the stochastic model, most of the time the tumor cells are heterogeneous and they can proliferate extensively. However, the second theory, which is the cancer stem cell theory, is also called as the hierarchical model. In the hierarchical model, as is seen, there is only one cell type which is represented here with a yellow ball, which can lead or give rise to the tumor population. Here, the tumor population is definitely heterogeneous. However, they all carry a characteristic cancer stem cell property. And these also have the property to extensively divide and form the tumor. So what is that differentiates a normal stem cell from a cancer stem cell? Or we can ask the question, how the cancer stem cell arise? The cancer stem cells actually arise from the normal stem cells following an injury, which can lead to either an activation of an oncogene or the loss of a tumor suppressor. Further, it can have a deregulation in various signaling events leading to uncontrolled cellular proliferation, leading to the process of carcinogenesis. Let us now understand in the next slide what all different pathways exist in the origin of the cancer stem cell. It is interesting to note that the signaling pathways are important in not only maintaining a normal stem cell, however, deregulation of certain key pathways are also important for maintaining the uncontrolled proliferation of a cancer stem cell. This table here shows the important pathways which are important in maintaining both the stemness as well as the cancer stem cell property. And these pathways include the Wnt signaling, the BM1 signaling, the Notch signaling, the sonic hedgehog, etc. For instance, the Wnt signaling pathway is important in maintaining the hematopoietic cells. However, a chronic Wnt signaling is associated with cancers such as the leukemia. Similarly, the sonic hedgehog, when regulated, is important for hair follicle progenitors. However, a deregulated sonic hedgehog pathway ends up leading to basal cell carcinoma in the skin. There is a thin line between the process of normal regeneration vis-a-vis that of the cancer. The reason being, both normal regeneration as well as cancer requires proliferation. However, in a normal regeneration, the proliferation is controlled. However, during cancer, it is uncontrolled. This slide explains, on the basis of a normal and a cancer stem cell, the process of regeneration versus cancer. As an example, in figure A, you have a layer of resting epithelial cells, which show different cell phenotypes, the brown, orange, and the yellow cells. And all these cell types in turn are derived from the tissue stem cell or a progenitor, which is shown in red. Most of the time, the tissue stem cell or the adult stem cell is quiescent in nature, that is, it is not dividing. However, following an injury, pathways such as the hedgehog and the Wnt signaling become activated. This causes the red-colored stem cell to become active and help in the process of repair and regeneration, and thereby filling in the injured area. Once the injury is healed, the repair process of hedgehog and the Wnt signaling pathways cease to become active. This stops the process of the regeneration. Now let us understand what happens during a chronic insult or a chronic injury. During chronic injury, there is a continuous activation of the hedgehog and the Wnt signaling pathways. And this leads to continuous or uncontrolled division of the stem cells. As an example, with the activation of this pathway, there is an accumulation of the reactive oxygen species, which in turn can damage the normal stem cell, and the normal damaged stem cell can then result into a cancerous stem cell, which can then initiate the process of cancer. So students, now you realize how both regeneration as well as cancer have some pathways which are common. However, these pathways work differently in context of regeneration versus cancer. Continuing with the signaling pathways, let's understand how the signaling pathways dictate different cellular fates. Here is an example of hepatic injury and how the cell fate can be dictated towards a biliary cell type or a hepatocyte depending on the signal pathway activated. Whenever there is a hepatic injury, the progenitor cells get activated. A progenitor cell with an activated JAG or a notch pathway leads to a biliary cell fate, and more increase in the JAG1 ligand can turn the biliary cell towards cholangiocarcinoma if the proliferation is uncontrolled. On the other hand, if the Wnt and the beta signaling pathway gets activated, then the progenitor cell fate is driven towards hepatocyte regeneration. Usually, the regeneration process stops following inhibition of a Wnt signaling. However, if there is an activation of the beta-catenin due to mutation, then instead of normal regeneration, the hepatocytes continue for uncontrolled proliferation, thereby becoming malignant, leading to the formation of hepatocellular carcinoma. One important concept both in stem cell as well as in cancer stem cell is the concept of niche. So now let us understand what is meant by a stem cell niche. A stem cell niche is actually an area within the tissue that provides a particular microenvironment. This microenvironment in turn is dictated by the presence of the various cytokines or the chemokines existing in the milieu. This microenvironment helps the stem cell present within that particular area to remain in an undifferentiated state, and also it can help in the property of self-renewability. Cells of the stem cell niche interact with the stem cells to maintain them or promote their differentiation also. Let us understand this with a diagram which is shown in the next slide. This is a graphical view of a human colon intestinal epithelium, and it shows the presence of a red area which is the stem cell niche at the bottom of the crypt, and this is where the stem cell exists. As you can see, during the proliferation, there is a difference in the compartmentalization of the proliferation component as well as the differentiation component. Further, the stem cell niche is dictated by the presence of the different growth factors and the signaling pathways which are operating in that particular niche area. In a classic work by a Japanese scientist, Yamanaka, he and his group discovered 4 factors, namely OCT, MYC, SOX, and the KLF, which can transform a normal fibroblast into a stem cell-like cell. This is a process of induced pluripotency. and the cells thus derived are called iPSCs. For this work, Yamanaka was awarded the Nobel Prize in Medicine. So now students, you can realize the importance of this technology because any cell in the body can be taken and it can be transformed with these four Yamanaka factors to derive a pluripotent stem cell. Let us now understand what all are the different markers that can help in identifying a stem cell and a cancer stem cell. The four important factors which can help as markers to identify a human embryonic stem cell are the OCT4, the NANOG, TRA1, SOX2, and SSEA4. This slide shows how one can identify the cancer stem cells by the flow cytometry method. In flow cytometry, a site population is a subpopulation of cells that is distinct from the main population. Site population, at least in case of the stem cell, is defined as those cells that show higher efflux of the DNA binding dye host. Because of high efflux, the site population or the SP cell appear dim. The molecules involved in effluxing the drug are members of the ATP binding cassette family, such as the MDR1. And in fact, this is a property which is special not only to the stem cells but also to the cancer stem cell. Because of the high expelling property of the anti-cancer drugs, the cells have improved survival and sustain their clonogenicity during exposure to cytotoxic drugs, thereby allowing the disease recurrence. Thus, identification of a tumor progenitor, or for that matter, even a stem cell population, is by the intrinsic mechanism for cytostatic drug resistance that might provide clues for improved therapeutics. The other way that one can use to identify the cancer stem cell is by the use of the cell surface marker, and this is shown in the next slide. This slide shows the important cell surface markers which can be used in conjunction with either flow cytometry or by imaging to identify a cancer stem cell. The most important marker in this is the EPCAM. EPCAM is basically an adhesion molecule on the cell surface which is commonly present in certain cancer types such as the pancreatic and the liver cancer. In addition, the CD or the cluster of differentiation can also be used in differentiating different types of cancer stem cells based on their origin. For instance, CD34, CD38 negative cells mostly constitute the leukemic cells. CD133 is a common marker present in the brain, lung, colon, and the pancreatic cancer stem cells. In cancer treatment, identifying a cancer stem cell has both a diagnostic utility and also selective elimination of the cancer cell by chemotherapy will be of immense use in cancer therapeutic. As this slide shows, there are two kinds of balls, a blue ball, which is a normal cell, and a cancer stem cell represented by the red ball. The journey from a normal to a cancer is through uncontrolled proliferation of the cancer stem cell. And selective elimination of the cancer stem cell will have promising avenues for the chemotherapy. Dear students, today we learned the differences between a normal and a cancer stem cell. We learned how some properties like self-renewal and differentiation are common characteristics of both normal and the cancer stem cells. However, the rare cancer stem cell have uncontrolled proliferation rate unlike a normal stem cell. This module also discusses the hierarchical and the stochastic model of cancer cell origin. As per the stochastic model, almost any cell can form tumor. However, in a hierarchical model, only a rare cell population can give rise to cancer. Hence, the concept of cancers being derived by the cancer stem cells or tumor initiating cell population got originated. We also learned the role of stem cells in normal regeneration and how deregulation in key signaling pathways, such as that of the oncogenes, can transform a normal stem cell into characteristics of a cancer stem cell property. Further, we learned the different signaling events that determine the various cellular fates. This module also helped in understanding the concept of stem cell leash, which is nothing but the microenvironment where the stem cell reside and help in maintaining not only the undifferentiated phenotype, but when needed, it can also get differentiated. We learned how the stem cells can be identified using flow cytometry through the site population or through the presence of certain cell surface markers, which can be then used to find a cancer stem cell. One of the best examples is the EpCAM positive cancer stem cells in hepatocellular carcinoma. Lastly, we learned that identifying a cancer stem cell is important not only in cancer diagnostics, but also specifically targeting the cancer stem cells has a therapeutic potential. So, students, in conclusion, this module covered the cancer stem cells show a property of self-renewal as well as the property of uncontrolled cellular proliferation. We also learned commonality and differences in regeneration vis-a-vis cancer in terms of tumor initiation and properties of the cancer stem cells. We also learned what a stem cell niche is and the various markers that can differentiate a normal from a cancer stem cells. Further, the utility of the cancer stem cell identification is important not only in diagnostics but also in cancer therapeutics. Thank you.